Hello, welcome. So it's a great pleasure to have today in this fantastic liver seminar series Dr. Jan Boren. Dr. Jan Boren, he has a dual profile, so he's an MD and also a researcher. He's a MD in the hospital of University Hospital of Gothenburg. This is the pronunciation in Swedish, Gothenburg, and he's in the clinical medicine, molecular and clinical medicine the department. So his research mainly is around fatty liver, with the idea to understand not only mechanisms, but really look for therapies with really a strong focus to translate to the clinical practice all the discoveries. And he's using a system biology approach, and he will explain his exciting last results on this on the mechanism and consequence of fatty liver disease. Thank you very much, Jan, to be so kind to came to explain your research to us today.
Thank you very much for the very kind introduction and a very nice invitation to sunny Barcelona. When you come from Sweden, you really enjoy that, I can tell you that. It's dark when you get up and dark when you get home, and it's cloudy in between. The weather has changed, so we don't have any snow now. We have only rain during the winter. As Marta said, I work at the Sahlgrenska University Hospital in Gothenburg, and I'm the head of the Institute of Medicine, and I belong to the Department of Molecular and Clinical Medicine. Today I will talk about the mechanism for and consequences of fatty liver disease, and if you have any questions, just raise your hand immediately. As you know, the risk factor for cardiovascular disease have changed quite a lot. Some things are getting smaller, and some things are getting wider. But in reality, this has a major effect on how we treat and identify patients, because earlier we had mainly a hypercholesterolemia, whether smoking, hypertension. But the hypercholesterolemia is pretty easy to handle today. You have statins, you have PCSK9 inhibitors, but today we have a much more complex dyslipidemia or hypertriglyceridemia that we see in type 2 diabetic patients and obese patients. And I just want to go back to the primary or most important risk factor for cardiovascular disease, that is increased blood lipids. And we have two types of lipoproteins in the circulation. We have chylomicrons that are synthesized in the intestinal cells. They are very triglyceride rich. They transfer triglycerides from the diet out to the different tissues, and they unload the triglycerides by the action of lipoprotein lipase. And then these chylomicrons are then taken up by the liver. The liver secretes very low density lipoproteins, triglyceride-rich lipoproteins. In the circulations, the triglycerides are taken away from these particles, and we end up with a low density lipoprotein particle. This contains mainly cholesterol ester. And this is the main risk factor for cardiovascular disease. It's also called the bad cholesterol. The majority of these particles are taken up by the liver, and there is actually competition between the chylomicron remnant and the LDL particle. I actually tried, when I explained this, I over-explained, because we have two different forms of a VLDL from the liver. We have one form that is somewhat smaller, and this is the main particle. But in patients with type 2 diabetes, obesity, then we start to form this VLDL1 particle, and this is larger, contains more triglycerides. And this is in the circulation converted to VLDL2 and to the smaller particles. But why is this interesting? Well, if you look in the patient with type 2 diabetes, you can see the total plasma triglycerides, and you can see VLDL, VLDL2. The VLDL1 is in blue, and the VLDL2 is in green. And you can see that if you have 3 millimolar total triglycerides, the majority of these are carried in the VLDL1 particle. And as I said, this is really the most important predictor for the plasma triglycerides. And this is the one that is increased in patients then with type 2 diabetes insulin resistance. And we have a major interest to understand why. So what is really the underlying mechanism for this dyslipidemia? And the problem when you only look at the lab values, if you have 2.5, 3 millimolar triglycerides, you cannot really say if that is caused by impaired removal of lipoproteins from the circulation, or if it's due to oversecretion of triglyceride-rich lipoproteins. So to understand that question, you really need to perform dynamic studies, and these are then called kinetic studies, because then you can start to understand the mechanism that underlie the disturbed lipid metabolism in the patient. So what we do, we perform kinetic studies in order to understand both the secretion and the catabolism of these lipoproteins. These are pretty labor-intensive studies. First of all, you have to inject a range of different stable isotopes in the patient. And these patients are extremely well characterized. Then you take blood lipids, sample up to 96 hours. Then you isolate the lipoproteins using density gradient ultracentrifugation. And then we start to analyze the enrichment of the stable isotopes within both the lipids and proteins. And what you do is really you look at the enrichment of these curves. Here we see VLDL ApoB and VLDL triglycerides. And based on this enrichment data, you can use the output from those to use multi-compartment modeling. And then this is a simple model for VLDL secretion. You have the plasma here, you have the leucine pole that will go into label ApoB, you have the glycerol stable isotope that will go into the triglycerides in the liver, and then in the liver you then form lipoproteins that are secreted to the plasma. You secrete both VLDL but also VLDL2 particles. And using this modeling, you can then infer constants that will tell you about the secretion, turnover, and removal of the lipoprotein particles. And then you can start to understand what is really happening with this one. The problem is that each patient takes roughly 1 month to work through all the different steps. So it's a rather labor intensive. We have performed roughly 120 kinetic studies so far. So I think we have one of the largest population in the world. When we did this more than 10 years ago, we started to look at the different predictors for the oversecretion of VLDL1 particle, the triglyceride-rich particle from the liver. And at that time, we were somewhat surprised to see that liver fat was an important predictor, but also plasma glucose. At that time, to be very honest, we didn't pay too much attention to fatty liver. During more recent years, we have much more attention to this. But it really shows that hepatic overproduction of large VLDL is driven by hepatic triglyceride accumulation and hyperglycemia. What is then, why do we have an interest in fatty liver? Well, we all know that we see patients with a lot of increased adipose tissue. But the problem is not really when you store the fat in your adipose tissue. The problem is when you have impaired storage capacity within the adipose tissue. And this could be due to, both capacity to expand is limited or inflamed adipose tissue. There are several different hypotheses to explain this, and I think we can say that we still do not fully understand the underlying mechanism, why we start to store lipids in other tissues. But we do have an overflow of lipids, so we start to store the lipids in the viscera. in the liver, in and with around the heart, in muscle, or in pancreatic tissue. Just to underline this, it's not really the amount of fat that is important. Some of you might have seen these patients. These are lipodystrophobic patients. They have a mutation, so they cannot form adipose tissue. These patients do not have any tissue, virtually no adipose tissue at all. And then you might say, oh, they have no fat. They're healthy and nice. Unfortunately not. Because what happens is that since they cannot store the fat in the specialized adipose tissue, they start to store it in muscle, around the heart, in the liver, exactly as what is called an ectopic lipid accumulation in the obese patients. So these patients have fatty liver, they have metabolic dysfunction, very often insulin resistance or type 2 diabetes, despite being very thin, and they can look very healthy. But what is common within all these ectopic lipid accumulation predictors is that you will have increased risk of cardiovascular disease. And why? Well, when we have more fat in the liver, the lipoproteins contain fat. And when you have more fat substrate in the liver, you can actually form more lipoproteins, and you can load them with more plasma triglycerides. So you form the VLDL particle instead. So you have a roughly 45% increased secretion of triglycerides from the liver. But that's not the only thing that happens, because you have also impaired uptake of these particles in the circulation. Both the VLDL, IDL, but also the LDL, the cholesterol-containing particle. So since they are not taken up by the liver, you accumulate these particles in the circulation. So what is really characterizing these patients is that they have increased plasma triglycerides, they have accumulation of remnant particles because it's not cleared by the liver, they have small dense LDL particle, and they also have decreased HDL, that was earlier called the good cholesterol. So this is a really complex atherogenic lipoprotein profile in these patients. And what is problematic is that roughly 25% of all adults today have fatty liver. This is one of the most common diseases today. When we look in high-risk patients, obesity, type 2 diabetic, the prevalence is around 70 to 90% in many populations. It may progress to severe liver diseases like fibrosis, cirrhosis, and also liver carcinoma. But in reality, rather few patients will develop this. But since we have so many patients who develop fatty liver, we end up with quite a lot of patients. So, complications to fatty liver will, it's predicted, but it will within a few years be the most common cause for liver transplantation in the Western world. But most patients will not die from liver diseases. They will die from cardiovascular diseases. And the increased blood lipids, the atherogenic lipoprotein profile, is the main driver for this. There might be additional explanations because the fatty liver seems to be an independent predictor in addition to the dyslipidemia. That could be mediated by a pro-inflammatory state or other causes. So we are then interested in what is then really the pathophysiology of NAFL. What is really happening here? And in the first study I will tell you about, we took advantage of our population where we have extremely well phenotyped patients, where we have done imaging, so we know exactly how much fat they have. We have performed the stable isotope studies. We have performed a lot of metabolomics and lipidomics. So these are very well characterized patients. So we performed then kinetic studies, so we know the VLDL secretion in this. We have also plasma metabolomics and plasma lipidomics in these patients. The problem is then, for most people, you accumulate a lot of data. And sometimes I think we are exaggerating. I usually put comics as the last omics variant because in clinical medicine we collect so much data, but very few people know how to use the data, and eventually end up with this enormous Excel file, and then you sit there and see if you can find something. But today, of course, we have much more modern way of doing this, and genome scale metabolic models have made a major difference. And this is then a way of analyzing bioinformatics of the data. And I think this is very, very important, not only for people specialized in bioinformatics, but also for clinicians, because it really enables you to look at metabolic pathways, to understand what is happening, to integrate multi-layer omics data. So you will have added value really to understand what is happening, but importantly also to incorporate the clinical data that opens up the perspective of personalized medicine. And really, this is something we are very interested in, to get mechanistic perspective that guides feasible strategies for intervention. And there are several different models published. We very often use this iHepatocyte 2322 because Adil Mardonoglu, he's a good friend of mine, and we work very closely together. Adil got his PhD on Ireland, then went as a postdoc to Jens Nilsen's group in Gothenburg, and then became assistant professor at Royal Technical University in Stockholm, and he's now part-time professor at King's College in London. We have very close collaboration. One problem when you do very often do modeling is that you use population data, because you do very seldom have the individual data for every single patient. The strength with our way of working is that we do have quite a lot of data from every single subject, since we have the stable isotopes. So we have, in reality, we know the VLDL secretion in detail. We know exactly the amount of liver fat in these patients, and we have all the clinical data from our biobanks. And we collect a lot of clinical data and imaging on these patients. So they are, we were then able to combine our data files with a modeling system. And when we did that in this study, what we did was, use the genome scale modeling, and then we tried to understand what is then the rate limiting step in NFL patients. And after some work, we came up with this step. This is, here you have the mitochondria. Here you have the liver, you have the formation of glutathione precursors, and then glutathione, and then you have in the mitochondria an NAD plus production and fat oxidation. So in our data, we had an unbalanced redox state and increased demand for glutathione and NAD plus. That was what the modeling indicated. But then we started to test this. And of course, we did metabolomics, so we could look at all the plasma concentrations. N-acetylglycine, betaine, glycine, serine, they were all significantly decreased in these patients, indicating that we got some experimental support for the modeling. We also looked at several of these genes involved, and we did gene expression analysis from our data files, and we could see that these were then also decreased. So the data really indicated that this is a step that seems to be impaired in these patients. We had support from metabolomics, we had support from gene expression analysis. But is it correct or not? It's very hard to validate this in any way. The only way we can test this would then do to see what happens, and we try to increase these glutathione precursors in patients. So the first thing we did was, would it be beneficial to stimulate beta oxidation in fatty liver by supplementation of glutathione and NAD+ precursors? So we did first a very, very simple study. We frankly just gave subjects serine because, as you saw, serine was severely depleted in these patients. And we just gave serine and 200 milligram per day per kilo for 14 days. And the liver fat decreased from 27 to 20%. We had a placebo group as well. Nothing happened. So it indicates that we might be on the track to be correct. Why did we only use one precursor here? Well, the reason was ethics. We could very easily get a fast ethical approval to give serine to these patients. But you can see we also decreased ALT, SAT, alkaline phosphatase. So, indicating that we had improved liver function just by giving the serine. But then we went into mice, and then we gave mice 16-week, we induced liver fat, and for 2 weeks we gave a cocktail of glutathione and NAD+ precursors. We gave these precursors over 2 weeks. And this cocktail contained nicotinamide riboside, serine, N-acetylcysteine. And you can see that this is the treated mice, and these are the control mice in blue. And we looked at the liver triglyceride accumulation, and by giving this cocktail, we could see a 50% reduction in the fat accumulation over 2 weeks. We thought that was quite promising. We did not see a difference in different phospholipids. There was a trend in cholesterol esters and a trend in ceramides. You can see that we increased serine, we increased glycine, did not see a difference in glutamine. So it indicates that this could be beneficial. We also characterized the triglycerides in the livers of treated mice and control mice using lipidomics. And what we did was that we separated the triglycerides based on the length of the fatty acids that are attached to the glycerol. And the reason for this is that triglycerides with long fatty acids, they are not sorted to beta oxidation in the liver. It's preferentially the triglycerides with short fatty acids that will undergo beta oxidation. And we put the controls as 100%. And as you can see, it's mainly the shorter forms that are decreased in the livers of the treated mice, indicating, supporting that is enhanced beta oxidation we are looking at. But as I've discussed with Marta today, you really need to have more experimental proof looking at the beta oxidation, characterizing this at every step. What we have done as well is that we have applied for permission from the different agencies and ethical committees, and we just got ethical permission to perform a human clinical study using this cocktail. And there we have also added carnitines. We'll give nicotinamide riboside, serine, carnitine, and N-acetylcysteine. We will give this for 6 weeks. We will start for 2 weeks with a half dosage and then go up to full dose. And in all our studies, we do metabolomics, lipidomics. We measure hepatic de novo lipogenesis using heavy water. We use imaging of the liver, so we are sure how much liver fat we have in the other fat compartments. We also usually take quite a lot of biopsies. Here we're going to take adipose tissue and muscle biopsies and stool samples. So it becomes a rather complex clinical study. But we think it's important to try to do as much as possible when we do these studies. So this we are virtually just starting to recruit the patients for this study now. So it will be very interesting to see if the modeling that seemed to be positive in mice will hold up in patients. And that is really the profile we are trying to work along. I was also sitting with a very good friend of mine, and we're discussing several different mechanisms to induce fatty liver. Then we went for lunch, and when we came back after lunch, we said, shouldn't we discuss how to unload the fat from the liver instead of inducing more fatty liver? And we were both quite interested in a low-carb diets in different. So the question is, can we improve NAFL and hypertriglyceridemia by reducing carbohydrates in the diet? A problem you very often have with low-carb diets is that they lose weight at the same time. So we set up a study where we gave isocaloric low-carb diet. To do that, of course, you have to increase protein and fat in the diet. And what is the reason for this diet? Well, roughly 25 to 30% of all triglycerides in the liver in NAFL patients actually come from carbohydrates. Because you have what is called the de novo lipogenesis, where you convert glucose into fatty acids in the liver. And this is if you don't have fatty liver, this is down to 1 or 2% normally. So this is heavily increased in these patients. And one of the reasons why it is increased is that the enzymes that are needed for hepatic de novo lipogenesis, the expression of these genes is driven by both hyperglycemia and hyperinsulinemia as well. So in these patients where you have increased insulin, increased glucose, you have more enzymes, you have more capacity for de novo lipogenesis, and of course you have a lot of substrate. There is also one thing more that is important to remember, that normally you have beta oxidation in the liver, but when you have de novo lipogenesis, you generate something called malonyl-CoA, and metabolite. And this malonyl-CoA inhibits uptake of fatty acids into the mitochondria, and hence you block the beta oxidation at the same time. So these patients then have increased beta oxidation, forming more triglycerides, but at the same time, they have impaired beta oxidation in the liver. So what we did was that we took 10 middle-aged obese subjects. They had a BMI around 34 and the liver fat around 16. They were extremely well characterized. Then we served isocaloric, low carbohydrate diet, and we used less than 30 gram carbohydrates per day for 14 days. And what we did was that first 1 week we put them on, had dietary records. Then we produced all the food in a metabolic kitchen. We froze it down. And a nutritionist went home to every single patient every second day with more food. Everything in addition to what they should eat that was frozen, like tomatoes, she bought that for them. She gave them everything. Then she, on a daily basis, twice had contact with the patients, checking that everything was under control. We checked the weight twice per day. And these, I really love these people because they are so careful. There was one patient who had 1 kilo difference when she was in the hospital on the same day and when she checked her weight in the evening. And the dietitian figured out that the floor in her bathroom was slightly uneven. So that patient went out on her balcony every day for 2 weeks to check her weight. Because that was the only floor that was completely even in her apartment. These, we are very thankful to these people. But we were able to keep the weight. You always lose roughly 1.5 kilo because you have glycogen in the liver. And when you put the patients on a low-carb diet, you will lose the glycogen. And 1 gram glycogen binds 4 grams water. So you will and you have roughly 300 grams of glycogen in the liver. So you lose 1.5 kilo, mainly water. And then we performed, we checked using a spectroscopy liver fat 7 times over 2 weeks in every single patient. We used heavy water to analyze hepatic de novo lipogenesis. We looked at the body composition. We looked at markers of inflammation. We collected stool samples for metagenomics 4 times over these periods. We performed metabolomics. And in a second cohort of identical patients, we performed 2 liver needle biopsies. Of course, we didn't want to do that in the same patients where we collected all the omics data. Sweden is a very strange country. You would never be able to perform 2 liver biopsies in mice. We would never get permission to do that in mice. But you can get it in humans. There we had 7. I also want to really thank Hans Ulrich Marshall, who is the hepatologist. He's a fantastic hepatologist, and he collected the liver biopsies. First, as I said, the carbohydrates are converted to fatty acids via the hepatic de novo lipogenesis. And we used heavy water to analyze this. And you can see that the first day was on day 3. We analyzed the de novo lipogenesis 3 times. And you can see there's a dramatic drop in hepatic de novo lipogenesis, showing that you have a very quick response in the liver when you take away the carbohydrates. We also measured the beta-hydroxybutyrate as a surrogate marker for beta oxidation, and it was a 6-fold increase in these patients. Because when you block the de novo lipogenesis, then you block the formation of malonyl-CoA, and then you can have full speed on the beta oxidation. Then what happened with liver fat? Well, this is the group. We started around 16-17%, and the mean loss was 6.4%. The range was 3.5 to 8.8, and relative loss was 44% in 2 weeks, despite that they did not lose any weight. These are the patients, and I've done quite a lot of clinical studies, and you usually have quite a variation between different subjects. But here we did not see virtually any variation. They started up on a different level, but these curves are, as you can see, almost parallel. So they all lost liver fat as fast, with the same efficiency. And the relative loss, the range was from 30. Of course, when you start up here and lose 6.4, it corresponds to 30%. If you start down here and lose 6.4, it equals a 75% relative loss. So it really shows the importance of de novo lipogenesis. And VLDL triglycerides, we had a 58.1 decrease over 2 weeks, despite the fact that they consumed more fat. And plasma ApoC3, that is a important inhibitor of lipoprotein lipase, and it's an important predictor of plasma triglycerides. There was also a very nice decrease of this one. This is driven partly by a glucose regulatory element in its promoter. That's why you see a decrease. So this study really shows that the low carb, when you decrease the low carbs, then you have decreased de novo lipogenesis, increased beta oxidation, and decreased liver fat. But is this the whole story? We also have the gut microbiota. And we had collected samples over several times over this period. And this is, we sequenced this, and this is day 0, day before the change diet, day 1, 3, 7, 14. A very good friend of mine, I recruited him to the University of Gothenburg, is Fredrik Beckhed. He's one of the world leaders in gut microbiota. And he told me before that he was not really sure it was worth to sequence only 10 subjects, because quite unlikely you will see any change. But he's a very good friend of mine, so he did it anyway, and we were both quite surprised and amazed by the result, because you can see that you virtually change your gut microbiota in 24 hours when you change the diet. You have a very, very rapid response in the gut microbiota. And when I've seen this data, I do have to confess that I'm somewhat, I shouldn't say skeptical, but somehow concerned when I see people collecting a lot of stool samples and then they use that to predict risk for cardiovascular disease or type 2 diabetes in 5, 10, 15 years. I think you need to make sure that these patients have not been out to a different meal or gone to after work the day before or gone for vacation or something like that, because you have a very, very rapid change. And this is illustrated here when you can see that from day 0, day 1, huge difference, and between 3 and up to 14, quite minor changes. So it's a huge and very rapid response in the gut microbiota. Virtually all strains decreased. There are 3 that actually increased. Streptococcus, Lactococcus, Eggetella. And I will come back to why I pinpoint these a bit later. Then we started to do metagenome annotation, really to understand what is going on here. And we tried to look at pathways that started to change when we changed the diet. And there are virtually 2 major pathways, starch and sucrose metabolism, that decreased. And that's good. We put them on low-carb diet, so I'm happy that that pathway decreased. But to our surprise, we saw that folate biosynthesis increased already at day 1. And we did this by linking microbiota genome to pathways, the KEGG database. And we did a detailed KEGG analysis of folate biosynthesis, and 7 out of 12 were significantly increased. You can see them here. Really indicating that we actually, in the gut microbiota, we have more folate biosynthesis, because a lot of the folate we have in circulation actually come from the gut microbiota. And of course, is this clinically relevant or not? Well, I work partly in clinical chemistry, so it's very easy to test it. We just run the samples in clinical chemistry. And you can see that you increase the plasma folate from roughly 25 to roughly 35 nanomole per liter, just by changing the diet. And importantly, the diet we gave them did not contain more folate than the standard diet. We also used untargeted serum metabolomics to investigate how the bacterial folate affect host metabolism. And we saw that 202 out of 648 detected metabolites displayed significant changes and correlated with serum folate, serum biosynthesis pathway, and the folate producing. And I told you there were 3 strains that increased, and these are all folate producing gut microbiota: Streptococcus, Lactococcus, Eggerichia. So it really indicates that the bacterial folate plays an important role in human lipid metabolism. I can say that we also found metabolites indicating enhanced beta oxidation in mitochondria and peroxisome, in line with our surrogate marker for hepatic beta oxidation. And then we performed the liver biopsies, and we performed then 2 liver biopsies before and after 1 week on the low-carb diet. And we didn't keep these patients on 2 weeks. We said that we wanted to see the rapid changes and not the secondary changes to the first changes. That's why we run it 1 week. And we performed global transcriptomics analysis to identify differently expressed pathways. And then we looked we also, of course, used the genome scale models and plasma metabolomics. And this is what we see. We see that we have decreased glucose uptake as predicted. We have increased serine uptake. We have increased fatty acid uptake. We have increased fatty acid oxidation. We have increased uptake of branched amino acids. And importantly, we have increased serine, we have increased folate biosynthesis, and increased NAD plus production, and thus contributing to the fatty acid oxidation. And in line with the first study I showed, where we had impaired glutathione production according to the model, we actually in this clinical study see that we have increased plasma glycine and increased production of the glutathione precursors and more glutathione production, in line with increased beta oxidation. Just to emphasize the folate metabolism, you can see it here. Here you see nicely the conversion between the glycine and the serine. And the mitochondrial respiratory chain dysfunction leads to alteration in this one-carbon metabolism. And folate has a key role in regulating one-carbon metabolism, including the serine to glycine in the conversion. So these 2 studies fit nicely together, actually better than we had anticipated. There's a nice review by Ducker in Cell Metabolism on this topic. So, the data clearly show that, when you change the diets, put them in a low-carb diet, you have massive changes consistent with increased beta oxidation and increased one-carbon metabolism and more glutathione production. So the data shows that it's not only this low-carb decreased lipogenesis, increased beta oxidation, decreased liver fat. Also, gut microbiota is rapidly and dramatically changed when you do this diet intervention. And you have a more folate production, and folate is then linked to the beta oxidation, so you enhance the hepatic beta oxidation at the same time. Or all the data strongly indicate that. What is then the importance of this folate? Well, the serum folate could explain 35 versus 20% of the variation in liver fat before and after adjusting for BMI. So it seems actually to be a more important contributor than we had ever anticipated. but it clearly shows the rather complex responses you get after changing the diet, and also the power of bioinformatics systems medicine really to understand what is happening here, because all the data, we would not really been able to pinpoint and understand the full picture unless we had used the modern systems biology today. So that's why I think it's very important for also clinicians try to, utilize this, these technologies, or work with people who are really skilled in doing this. So the rapid metabolic benefits of a low-carb diet is that it improves liver fat metabolism. You see a rapid shift in the gut microbiota composition. It associates with increased circulating folate, and you have increased folate-dependent one-carbon metabolism in the liver. It improves all cardiometabolic risk factors we analyzed. Inflammation went down highly significantly already after 3 days. All plasma triglycerides, all lipid profile, we were surprised. And of course, it's a potential exploring diet microbiota interaction for treating NAFL. So this is, an important step. I really want to thank my close collaborators, people in my lab, Stefano Romeo, who did a lot of genome. When we picked up the samples, we excluded all genetic variants that are known to influence normal lipid handling. Johan Bergström, we have in Sweden something called SCAPIS. It's a national-wide screening of 30,000 subjects. It's a 1.5 day characterization. When we screen them with CT, we do all virtually ultrasound, we do virtually everything. So we are building a cohort of 30,000. It will be ready in a few months. It's the Swedish Heart Lung Foundation who has been investing in this. They have, I think they spent 35 million euros to establish this cohort. All university hospitals in Sweden are engaged in this, and they donated all the equipment, everything, to run this study. It was initiated and funded by the Heart Lung Foundation. And of course, now we have a lot of screened subjects at every university hospital, so it's very easy for us to pick up patients with fatty liver due to this. Hans Ulrich Marshall, who is the hepatologist, one of the few persons who can take liver biopsies, and the patient will ask, when will you do it? And he's already done it. He's a fabulous clinician. My mother-in-law, she's married to a clinician, and when she met him, because she had some problems, she said, I met the most wonderful clinician in my life. He's a good guy. Tony is doing a lot of bioinformatics together with Adil Mardanoglu, and Fredrik is responsible for helping us with gut microbiota. I have a very, very close collaboration with Marja-Rita Taskinen in Helsinki, Finland. We do a lot of studies together. Kirsi Pietiläinen she was really heading the team of dietitians, nutritionists, because she's an expert in this. Hugh Barrett and Gerald Watts in Australia, we've been doing, in the first study, some of the stable isotopes were performed in collaboration, also together with Bruno Vergus. We had intellectual input from Kevin Williams. So thank you very much. I can tell you, I could spend 2 hours discussing that issue with you. I've spent, long time discussing this with Kirsi Pietiläinen. And yes, the problem in this study, the weakness in this study, is that you do change the fat composition. Because in order to give the patients some food that is eatable, we were forced to give them, more unsaturated fat than saturated. Otherwise, they would not be able to eat it. And we thought it was better to give them something that was eatable, so they were actually sticking to this diet. And this diet was actually fine, because I had one freezer, we ordered more food than we needed. So all my postdocs and PhD students in the lab had this for a few months. It was free, of course. But you're absolutely correct. And along those lines, we have a close collaboration with a team in Maastricht. And there they have developed a methodology to analyze the lipid characteristics, saturated, non-saturated, using imaging technology. And we have done that study, and it's very clear correlation between saturated fat, of course, and liver fat accumulation. And we've all even been thinking whether we should do another study like this one and give different types of fat and then do the same omics characterization. So it's a very, very important question. In the best of all worlds, we would have had 2 arms with different fats, but we said that would become too complicated, because the imaging department in the hospital is not very happy when we do this, because over not too long time we have each patient underwent 7 spectroscopy, and since we had 10 subjects, we did 70 spectroscopies of the liver over a limited time, and they don't have that capacity in the hospital really to bring in more patients than that. There's a lot of regulation about the amount of folate you can give to humans, because it's a risk associated with the folate supplementation. We've been thinking about it, but the legal issues and ethics is a major hurdle for that.
Because
we picked serine only due to we could have taken glycine instead, but serine has been given to quite a lot of patients with a specific enzymatic defect, where they need to give them serine. So serine in high doses have even been given to pregnant women. And of course, it's much easier to get ethical permit when you can show that it has been given without a problem to pregnant women. So to have the ethical permit for serine was very easy. I don't think so. It could be, to my knowledge, I'm not aware of it, but I'm very often wrong, so I might be wrong here as well. Well, I heard the first question, and we can start with that one. You're absolutely correct, of course, that NAFL is not a clinical. The most important question is, of course, the NASH in the clinical scenario. I'm phrasing NAFL because in the first cohort we did not have liver biopsies on those. And of course, we would need liver biopsies to, put the diagnosis NASH on those. In the second cohort, where we actually performed the liver biopsies, there we did histopathology on those patients, and in reality, they had NASH, as expected. So the data, as what we have today, is that all the gene expression analysis, all the imaging in the second cohort was performed on NASH patients, and you see the beneficial response in those patients. In the first cohort, we do not have markers of NASH in those because we don't have liver biopsies. That's why I stick to the name NAFL. Very important question, and I can only refer to virtually the answer I gave for the first question. That in the second cohort we did have quite severe liver derangement in some of those patients, and that's why we were slightly concerned when we did the liver biopsies in those patients. When we looked at the gene expression analysis, even genes related to fibrosis decreased. All inflammatory markers, went down. So, we need to repeat this in a larger cohort of NASH cirrhosis diagnosed patients to answer that question. But I do have to say that I was a bit surprised myself to see that the NASH patients and with early fibrosis, and one had also closer to cirrhosis, that they all responded.
So in this case, we can close the session, thanking you again for your nice presentation.
Thank you.